Hello, everyone. Thank you very much for joining us in the talk from the Physio webinar spin-off, Advanced Topics in Thermobiology. This event was organized by graduate students from the Physiology graduate program of the Institute of Bioscience of the University of São Paulo. We organized very interesting talks that happened during 2 days with speakers from different countries talking about different themes related to thermobiology and the intersection with other areas. You can watch past lectures on our YouTube channel. Please follow us. They were amazing. After each talk, everyone can ask questions through the YouTube chat. They can also be in Portuguese if you like. And our team will select them and send them to me. This event will be recorded and will be available through YouTube after the transmission. As a first speaker, we are very pleased to welcome Dr. Carlos Navas, a biologist by the Universidad de los Andes in Bogotá, Colombia, PhD in Ecology, Evolutionary Biology, and Ecophysiology by the Universidade of Connecticut in Storms, USA, and today is a full professor in the Department of Physiology in the Institute of Bioscience at the University of São Paulo in São Paulo, Brazil. His research is about the impact of environmental change on organism performance using tools derived from classical ecophysiology and in interaction with other disciplinary aspects. Environment-organism relationships, particularly in the context of ectothermic animals, in relation with temperature, hygrometry, and energy. Dr. Carlos Navas, thank you very much again for your presence here today. After your talk, we will have 5 minutes to take questions from the audience.
Thank you, Maiara, Gustavo, all the staff that put together this seminar. I'm going to share my screen. Please let me know when this is okay, when you can see my slide, please.
We can see your slide.
Thank you. So it is a pleasure to be here, and it is also nice to see that some of the things that I will discuss today have some links with issues that were cited in the talks yesterday. What I'm going to do today is to discuss some of my findings in this collaboration with Volker Loschke and Gustavo, who is organizing this event. And it's about volunteer exposure to extreme temperatures in fruit flies. I'm going to spend more time explaining today the whys about this research more than the results, because I think it is important to describe in some detail what happened here in that context. This research that I'm going to present today had three lines of influence. One is represented here by one paper, Nelson, Heath, and Prosser. Heath and Prosser particularly were authors like extremely influential in my academic life. And in this review paper in American Zoology, they proposed that perhaps the first type of thermoregulation to evolve was the avoidance of extreme temperatures. And they proposed that this first step was probably influential in all the derivations of thermoregulatory behavior that emerged after. In the late 70s, Hutchison and Maines and Victor Hutchison, a researcher that I met several times and which was also very influential, probably pioneer on the studies on high elevation amphibians. They put together this paper that put me to think a lot, in which they described physiology and behavior as two aspects of the phenotypes that were so linked that they could not be easily differentiated. And they say that because of the possibilities of hardening and modification of thermal physiology, because of the thermal history of individuals, the behavior of individuals would be both cause and consequence of physiology, particularly thermal physiology. And they point out by the time that eventually behaviors could evolve that promoted exposure to critical temperatures. And I got a more recent paper as an example, because obviously those papers here are not the only ones that dealt with these topics. There's just examples that I brought about. But I got this recent review about something that I've been reading a little bit about, and it's the biology of invasive species. And there is a substantial body of literature at this point that talks about differences in cognition, particularly learning ability, the inclination to explore new environments, and the like. And the proposal is there that these behaviors associated to boldness could be represented in populations and favor the colonization of diverse environments. In the meantime, and here I get another review, if you check the literature specific on fruit flies, you will find already a very important group of papers discussing behavioral individuality. And all these papers generally agree that you have both genetic factors, environmental factors, and also what has been called stochastic factors, for example, neural wiring, all of those causing variation in the behavior of fruit flies and perhaps insects, and perhaps even in a more general way to other lineages. So what do we do then with all this information and these influences when we discuss thermal performance curves? One thing that is not treated so often in the literature is how this curve relates to behavior. Probably most people on our audience are familiar with these curves, but if not, those are general curves, hypothetical in this case, relating one performance function, for example, locomotion, speed, digestion, whatever you want, as a function of body temperature. And even if each trait study has its own curve, a general tendency is this, and you have a critical maximum temperature, a critical minimum temperature, and a range for optimal activity. So the question is, how do I expand this to behavior? So if I imagine in this intellectual exercise that this X axis of the figure is transformed into a thermal gradient, what should I expect? Should I expect animals then to range only on this maximum performance thermal range, or to explore the full range, even if facing suboptimal environments, or even facing the possibility of critical temperatures, even death? And you could imagine then that this behavior could range between a conservative strategy, represented here in yellow, to a bold strategy in green. And they would have both costs and benefits, according to what I just said. And a conservative strategy would be undoubtedly related to survival, but a bold strategy perhaps could be related to enhanced thermal adjustment and even to ecological opportunity. And Given this, we decided to engage into an exploratory study. And the main question was very straightforward. Would fruit flies face temperatures able to impair their behavior or even kill them? And those would be fruit flies that would not be in immediate danger or immediate need to get into this exploration. And we added several variables. For example, if the answer is yes, would this exploration be equal on cold and warm extremes? Aspects of, like hunger, for example. Fasted flies eventually would display like more remarkable exploratory behaviors. And if we compare different genetic lines, would we find differences? So all this was the exploration that we ran. But the first thing was actually to put together a system to be able to run those experiments. So in this paper that you can see below on my screen, we described both a system that we designed and we applied to ask these questions. So it's very important that you understand the system, because it is an essential part of this research. So here you can see the scheme is a drawing in which you basically see that you have three containers, one in which the flies start the experiment, one stopper is removed, flies can get up and choose to move into this gradient, and they will face one very hot spot and one very cold spot, and they may choose to stay within this part of the system that is less critical, or they may try to attempt a cold cross or a warm cross, and if so, chances are likely that are high that we can retain the crossing flies into these models, which are those that you can see in the real picture. Of course, we presented this system with all the technical details, as it's always necessary when you report an equipment for the first time, and all this is in the paper in a supplement. If this system is connected to thermal baths, as we did with water running into this silicone tubing, we can change the temperature and produce the gradients that that I just discussed. So this would be the cold side, this would be the warm side. There would be an amplification here. And this is the range of temperature. So you would see that in the in the warm side, in the very middle, we have temperatures at the beginning of the experiment, about 45 degrees Celsius, and temperatures about 10 degrees Celsius in the cold side. So the flies would actually face temperatures that would impair their behavior, this side, and would kill them, this side, if they stay long. But if they cross very rapidly, eventually they can manage to cross. So the approach, very briefly, was to start with near critical temperatures, to make the temperatures more moderate, but at a very slow rate as the experiment progresses. Count the number of flies that performed crosses, and then end the experiment by a fixed number of flies crossing or by time. And this was always with a rather fixed sample of flies to start with. And we compared some isolines and also fasted versus non-fasted flies. Our most important result is actually clear in these figures, when you can see all these flies with legs up. So the main thing was flies did try to cross both cold and warm sides. Some flies did die trying these crosses through the hot side. But a number of flies actually managed to perform very successful cold and warm crosses with different behaviors that I will not have time to discuss here. In this figure, just to present some of the results very rapidly, we have the total number of contacts with the critical zone. That means those are the flies that approach the critical zone, but not really necessarily trying to perform a cross. And you can see that this behavior is expresses more frequently at the beginning of the experiment over the first 20 minutes in particular. I don't think this is a video, but probably it's not going to work very well in the mode of reading that I am. But anyway, what you would see would be the flies ascending here from the test tube, and then moving into both areas of the system, eventually getting into this area and then performing a sharp U-turn. And this would happen both sides, and eventually, this is a short video only, so I don't mind not to show it. Eventually you would see one or two flies actually getting into the area of influence of the silicone. Oh, okay, so here we go. So I'm just to describe, this is the hand is removing the stopper, and then the flies are free to move, and they tend to enter, and they come back, and eventually we got already one fly that decided to continue, and that's what the video shows. Okay. Regarding the comparison of flies exposed to mild fasting, that was less than 10 hours of fasting, we have here the cumulative cold crosses and the cumulative warm crosses as a function of experimental time. And what you can see is that fasting did have an influence in the expression of this behavior, particularly regarding warm crosses, but it was not essential. That means not fasted flies did try to perform those extreme crosses. So the behavior is there, is intrinsic to the flies, and eventually can be modified by physiological states. In this figure, I have three isolines. Those are lines of fruit flies that are derived from one single female, so they have depressed genetic variation. We learned that in all these indicators, but perhaps the most important is this one, is the total crosses performed with experimental time, that isolines overall are depressed in terms of this behavior, even if some of the isolines express this behavior more than others. And this was an exploratory study in which we wanted to deduce if genetic components would be important. And that's what the data actually tell us, that you find both individual variation and genetic components, as it would be expected for behavioral traits in Drosophila. We made some experiments with flies that had already performed either a cold cross or a warm cross, trying to explore how repeatable was the data, and also to learn if flies performing crosses would be actually more prone to perform a second cross. This data was kind of confusing, but it does show one pattern. Here we have the flies that had performed a cold cross and before, and we see that if we compare those flies that had performed a cold cross with those in red that had performed a hot cross and a control in black, The previously culled crossers had more chances to perform a second cull cross than any other group compared. This was not the case for the one cross, but it does seem that the variation in behavior was quite increased in flies that had performed a cross either hot or cold. So this led several questions about repeatability, about inheritability, and other issues that were natural derivations of this exploratory data, and that actually Gustavo addressed later in the laboratory. So concluding, if we go back to this performance curve, what we find is the following. If we analyze fly by fly regarding behavior and this performance curve, we're going to find lots of variation. Some individuals will be more conservative, so others would be bolder. But if we translate into a populational analysis, what the population does as a collection of individuals, we would have any possible variation, and we would have a small fraction of individuals that is actually bold and explores either side of this curve, not necessarily both in the same individuals. And if we go back then to our original questions, yes, we found that fruit flies perform this volunteer exposure to critical temperatures, that this happens in both cold and warm environments, that this behavior is rare, but not critically exceptional. It's about 10% in flies of the wild type and much less common in genetically depressed isolines. And that the internal state of the fly, for example, hunger, may affect this behavior, but that hunger, for example, is not essential to elicit it. And I didn't have time to mention this, but briefly, we also ran physiological studies like the maximum and minimum critical temperatures of flies that had performed crosses, and we did not find any pattern that could easily explain behavior in terms of tolerances. For example, the flies able to tolerate the highest temperatures are not those that attempt more warm crosses. And that's basically what I have to say. I need to thank people who were absolutely fundamental for this project, and all the institutions that help provide an infrastructure, support of any type, and of course, financial resources. Thank you.
Oh, so thank you very much for your presentation. And we have some questions. The first says, does the high or low temperature in the experiment prevent it or make it difficult for flies to perceive the food at the bottom of the tube?
Yeah, we don't know about this. The system, because of the physical properties of the system, you find, for example, a humidity gradient, such way that you have more humid environment on the branch of the T system in the upper part of the apparatus. It is asymmetric in terms of humidity, and we don't know how this could affect the perception of flies regarding food. We suppose that anyhow the smell of food permeates the food system, and we don't know if case you could correct difference in humidity. Eventually you would have results somewhat different. If they are, perhaps they would be more in proportion, but not in yes versus no, because independently of these physical constraints of the system, both sides were explored.
Okay. The second question is, if the fly doesn't notice the food, does it just stop in the middle? Have you already quantified if the temperature influences the odor of the ingredients? Because normally these flies eat something like a fungus. So does temperature influence the odor of the ingredients?
Yeah, I mean, well, I have no doubt that the quality of the food could affect the results. But again, I say this in terms of proportions, not the main findings. And the other thing is that this is mostly about individual behavior. That means some flies individually would never approach the extremes. So it's not really what flies will do, but what individual flies do. So we study the population of flies in the experiment, but we pay attention to the differences in the individual behavior. Some of the flies, they do tend to explore a lot, and some of the flies are very conservative. So it would be more like an individual behavior. And within that individual consideration, the answer would be yes and no, in the sense that some of the flies do stay in the middle, and some of the flies do explore the full system, and the ones engaging in the second behavior are far more rare than the first ones.
Third question. According with your data, could climate change influence the proportion of wild flies resistant to adverse environments?
It is like a really long step before we can connect this data with, for example, ecological modeling and the like. But there is a link already established, and I think it's an important link, and is that at least the modeling based on mechanistic variables, for example, those that relate to physiology, they tend to propose thermal limits. And the model behaves as a barrier. That means if a critical temperature is present in the modeled world, then individuals would not cross. And what this experiment shows is, well, this is not that black and white. Eventually, behavior may make things more fluid, and eventually some modeling is more conservative than real life. This is to be explored.
Fourth question. What could be the role of social environment in thermally bold behaviors? Would you expect the same patterns testing individual flies instead of a sample of flies? How could you transform thermal boldness into an individual measure variable?
Yeah, this is something to think about a lot. And we are not there yet. Our study, as performed, it adopts a kind of a dichotomical position, conservative versus bold. And of course, this is an approximation. And what we have in real life is a full continuous between flies that are extremely conservative and others that are extremely bold. And within those extremely bold are some that actually died in the experiment, that were so bold that they stopped in the hot area and died. And so all these continues exist in the population. The questions then would be why this behavior is preserved by natural selection, and how could we study this in the future? And I don't have any doubt that this could be and should be somehow reduced to a behavioral index that would be a continuous, not a yes versus no. We are just not there yet, but that's an important observation and an interesting idea to pursue.
I have a question on my own, more technical. All the flies were from the same lineage, or you have a bunch of lineage just jumped together?
No, I actually didn't mention that, which of course was absolutely essential. I just forgot to mention it, but this was a highly controlled experiment. So it's a wild population in the sense that it's a laboratory population that's derived from an ample sample of flies collected in the field, and the experiment was very controlled. That means the age of the flies, the number of flies in the system, and all those variables could be important. I just realized that I didn't mention something related to the last question, and it was probably the core of the question about the social behavior. And so I forgot to say, I'm sorry, I have to go back to that question, but we don't know the social, we know that social interactions affect behavior, but we performed the experiment with a fixed number of flies, and we have no idea if we had worked with 10 flies or 100, if we had different results. My expectation, again, is that you may have different proportions in the finding, but that the core findings would have not been different. But of course, those things you need to do if you want to know for sure. Sorry that I forgot to mention that.
And now I think is going to be the last question. It's beyond my question. Question 5. Would you expect thermally bold behavior occur also in ectothermic vertebrates with high cognition capacities?
I think so. Eventually, not manifest exactly in the same way, but there are hints that animals in the frontier, for example, of invasion, they have different cognition, different behavior, and more risky behaviors. I'm also sure that this is influenced by natural history. I mean, I just don't see these sort of behaviors occurring in animals that reproduce at very low rates and that take a long time to mature. I think it's more likely to evolve in lineages that reproduce in very large numbers and have rather short lifespans. But as a general concept, we think that this is compatible with the notion of boldness that has been detected for invasive species independently of lineages. That means like a more aggressive behavior in terms of exploration, and that has been reported both in the context of interspecific comparisons, but also within populations when you compare individuals, for example, in central parts of the population and on the border, on the expansion border.
Oh, that's very interesting. Thank you very much, Carlos. We all thank you. And right now we are going to have, it's just a second. Here's something. Right now, we're going to have a 5 minutes break to welcome our another guest. And please, everyone, thank you very much, Carlos.
Thank you. Thank you all for putting together this event. I was very happy to be here today, and I congratulate you, Gustavo, and everybody on the Physio webinars, because this is like an excellent initiative. So thank you for being there and promoting these sort of events. And thank you, you all in the audience.
Thank you. Now we are very pleased to welcome Gustavo Agudelo. Agudelo-Cantero, a biologist by University del Valle in Cali, Colombia, and a PhD student at the Department of Physiology at the Institute of Bioscience of the University of São Paulo in São Paulo, Brazil. His research is about ecological and evolutionary response of ectothermic organisms to global changes. Current research is the relationship between thermal variation and the predictability of heat tolerance of larvae amphibians, ecological and evolutionary implications of thermal boldness. Gustavo Agudelo-Cantero, thank you very much, not only for your presentation today, but for organizing this event. After your talk, as you know, we will have 5 minutes to take questions from the audience.
Well, thank you, Maiara, and thank you to all the Physio webinar organizing committee and the staff for setting up this amazing event. Without your effort, this wouldn't have been possible. So thank you very much. I will share my screen now. Please let me know if you can see my presentation.
Yes, we can see your presentation.
Okay, thank you very much. Well, this research furthers what Carlos already presented about thermal boldness, sorry. And we here explored and tested actually the heritability of navigation behaviors of Drosophila melanogaster in extreme thermal landscapes. So an important thing to mention is that with very few and clear exceptions, most environments on Earth exhibit thermal variation, both in time and space, environmental thermal variation. And the thing is that a diversity of organisms of different body sizes, life histories, and evolutionary backgrounds experience a range of temporal and spatial environmental temperature variations. In this regard, it's important to mention that at least in motile forms, the first line of response would be behavior. We have seen through several talks during this event that animals integrate both behavior and physiology. And actually these names are not separated in the individual. But what we know is that for animals that with movement capabilities, their first response would be behavioral before some physiological effect occurs. And there are several ways through which thermal changes can affect behavior. And this is illustrated here. We have temperature changes in the bottom affecting behavior through different ways. The first way is what we call bottom-up kinetic effects of temperature and some top-down integrated effects of temperature on animal behavior. Regarding bottom-up kinetic effects, they act through different time scales of environmental temperature variation. And through physiological state, they affect performance of behaviors, as Carlos showed regarding, for example, the thermal performance curve, right? Changes in energy metabolism, changes with acclimation during within the lifetime of the individual, but also irreversible acclimation during development. All of these affect the rate, the velocity, ultimately the performance of behavioral functions. But also behavior can affect, temperature changes can affect behavior more directly through top-down integrated effects. And here, the neural system and thermosensory receptors on animals integrate information from the both of the temporal and spatial variation in environmental temperature, affecting the capacity of animal to make choices, to orient themselves and to navigate in spatio-temporally thermal environments, right? But also changes in behavior have the potential to affect body temperature through behavioral thermoregulation, taking advantage of a spatial variation in operative temperatures. Regarding all of this, we have seen that thermal behaviors span more than what we could expect from an optimal paradigm. For example, for animals looking some sort of optimal temperature, optimizing performance, or avoiding thermal extremes. There can also be thermally bold behaviors, right? As Carlos showed, I will not describe this in detail. We observed that the capacity of flies to cross through critical thermal zones, either cold or hot, they vary among fly lineages. Here in black, we can see on the left, we can see the number of cold crosses. In the right, the number of hot crosses through the experimental time. In black, an outbred population of Drosophila and isofemale lines in color. So this among lineage variation potentially suggests a genetic component underlying differences in thermally bold behaviors. But when we observe fly behavior in these experimental thermal landscapes, we saw that flies do actually so much more than just crossing or not, right? And thus this is a choice that underlies an exploratory drive. And that is why what I had the opportunity to research during my stay abroad at Aarhus University. So this is a model describing navigation behaviors in the system, in which if we put flies here in this home bottle, they will face two choices. The first one regards to the whether flies explore or not a new environment. Flies spend some time in this bottle before the stopper is removed, and then all flies can either go up to explore this new thermal environment, or they may stay in the bottle and never leave the bottle, right? And this is a decision faced by all flies in the sample. And this is what we have termed, and we have borrowed some terms from animal personality studies, and this is what we have called avoidance or exploration regarding novel environments. But then for flies that went up to explore this thermal gradient in this T labyrinth or system, they may either stay there, or they can actually cross a critical thermal zone, either cold or hot, and display thermal boldness, right? And that is the second decision, only available for flies exploring the system. They can exhibit either thermal boldness or shyness. This is a decision regarding engaging in a thermal risk. So our working hypothesis was that there is a heritable genetic basis underlying intrapopulation variation in these navigation behaviors of flies in experimental thermal landscapes, and specifically regarding risk-taking decisions. Thermal shyness is expected to dominate the response of flies that went up to explore the system under the paradigm of behavior risk conservation. We also asked whether flies display sex differences in their navigation behaviors across thermal landscapes. And whether navigation behaviors correlate between thermal landscapes, looking for some context dependency of how flies move around these environments. So what we did was to take advantage of the thermal decision system that Carlos fully described to set up two different kinds of thermal landscapes, different from the original design, in which we isolated both, we isolated cold and hot temperatures. So it's practically the same arrangement, just a water, only a water bath which circulated water, cold or hot water through the system, and we may see huge thermal changes on very short distances here. So we worked with 25 now isofemale lines of Drosophila melanogaster, but we worked with sets of 5 isofemale lines at a time. We controlled the density of flies during development in order to breed and produce virgin flies to work with. Flies were sorted and separated by sex under CO2 anesthesia. And we let flies to recover for some days and tested when these anesthesia effects had passed. So our experimental unit was this fly sample, this bottle that was composed of virgin males or females from the same isofemale line, that shared the same developmental environment and that were tested separately. So our ideal experimental design involved to get at least 3 of these fly samples per sex, per each isofemale line to test in each thermal landscape, right? And when we tested was fly behaviors. In what way? We performed 2 experiments per day during the light phase of the flies. We performed 2 experiments separated only by 30 minutes in order to test both sexes of these sets of 5 lines at a time, to test these sexes separately. So we randomly assign sexes to either of the first or second experiment during the day. And every 10 minutes, we observed and counted the number of flies in feeding bottles, in the lateral bottles, after crossing the critical thermal zones. And we counted the number of flies in the T system here that were represented here in the T system, but didn't cross. And from these counts and taking advantage of the known number of flies that we put in the bottle, I mean the initial number of flies, we calculated the number of explorers as the sum of the 2 counts here, the number of avoiders, that it was the initial number of flies that we put in the sample, minus the explorers, and the number of bold explorers and the number of shy explorers, right? So some of the results is that when we observe fly navigation behaviors in a population context by sex and in each thermal landscape, we can find these distributions, this occurrence of the behaviors. So in the x-axis, we can see the proportion of the fly sample. This is each dot here in the block spots represent 1 fly sample. And this proportion is the proportion of flies that exhibited a given behavior. So if we look, for example, to the left panels, the cold thermal landscape, and according to what behavior and decision we are looking for example, for females, let's compare the red and the blue distributions in the cold in the left here. We may see here that the median proportion of the fly sample that exhibited both avoidance or exploration was roughly 50-50, right? So half of flies in a sample were up exploring the system and half were stayed in the home bottle. But if we looked to the same data for males, we can see here that more males prefer to avoid exploring the system, and less males in median preferred to or opted better for exploring the system. When we compared second decision behaviors, boldness and shyness in the same thermal landscape, we can see for both sexes that the difference between boldness and shyness is balanced in favor of boldness. I mean, more flies within this population in the cold thermal landscape in both sexes opted for exhibit thermal boldness instead of shyness. When we go to the hot thermal landscape in the right, we can see that the differences are bigger. For example, more females tended to explore this system than staying in the home bottles. Males were opposite. More males tended to avoid the system, but those that were exploring opted for thermal boldness in both sexes more than shyness. Actually, shyness exhibited a very right skewed distribution. When we isolate these patterns for thermal landscapes, we can confirm through a statistical modeling that actually the relationship between avoidance and exploration that accounts for these first decision behaviors depends on the time we perform experiments. Let's see, regardless of the sex, flies tended to explore more. Here in this, the first experiment is this one, from 1 to 2 hours in the light phase of the flies, and there is a higher proportion on average of flies that explore the system compared to the flies that avoided it. But this relationship was reversed during the second experiment, separated only by 30 minutes, right? When we observe the proportion of flies that were exploring the system and only those flies that exhibited boldness or shyness, we confirm that regardless of the sex, flies exhibited higher boldness behaviors than shyness. But when we see the hot thermal landscape, we can see also that time of the day was a factor that affected thermally bold behavior, avoidance and exploration. And this time, females and males displayed differences. So regardless of the time of the experiment, females always tended to be more explorative than males. But in the second experiment, the relationship between avoidance and exploration was reversed. It was like the second experiment, flies tended to be less, to explore less in both sexes. When we see boldness and shyness differences, the sex was not important, but the time of the day was. During the first experiment in the left, the difference between boldness and shyness were huge, with on average 75% of all explorers in a sample exhibiting thermal boldness. This is crossing through the hot thermal, critical thermal zones, but this difference was reduced and still boldness was higher during the second experiment. When we compared the among line variation in navigation behaviors of fruit flies, and we consider these thermal landscapes differences and sex differences, we can see here how the height of the bars here displaying the average response within each line varies among lines, right? We see some phenotypic variation. If we see quickly from here, we compared color like red and blue bars within each isofemale line. It might seem that sex differences are not quite important describing these patterns. It's more among line phenotypic variation. But when we partition this phenotypic total variation into that variation associated only to the fact that lines were different, and if we assume that these differences among lines are basically genetic, we may see different patterns between thermal landscapes. In this table, we can see here variance components here. VL is the variance component related to lines, and the variance, the residual variance, the environmental variance, and all uncontrolled factors. And for the cold thermal landscape, we can see for most behaviors that the variance among lines is quite small, with low confidence limits close to 0, which led to a small and non-significant heritabilities and small evolvabilities. This is the the potential for adaptive evolution to occur, with the exception of thermal boldness of males. But this is in the hot thermal landscape occurred the opposite. We observed that for most behaviors, but thermal shyness of females, the variance among line was high, which led to high and significant heritability of these behaviors in the heat, and high potential for adaptive evolution to occur. So concerning the relationship between navigation behaviors between these thermal landscapes, phenotypically or genotypically, we didn't find any, right? And this is again because the variance component associated to lines, but also this time the genotype by environment interaction was close to 0. So most variance here was residual, which meant that there are a lot of things happening that we didn't consider in our design, explaining these differences. But the genetic correlation coefficient, even though in some cases was high, was not significant. And this has many implications. So regarding our hypothesis 1 about the heritability of these behaviors, we partially confirm it because we only saw that for the experiments in the heat. And this contrasts with evidences on the physiological component of heat resistance, with Drosophila and other ectothermic lineages has shown low adaptive potential. We saw that regarding adaptive potential of navigation behaviors in the cold, which means that there is a consistent response among lines, but thermal boldness of male in the cold could be still important. And the absence of correlation, of genotypic correlation precisely in behaviors between the thermal landscapes, can indicate that these are different traits. These behaviors are different traits governed by independent pool of genes, that there is no pleiotropy, for example, acting or controlling these behaviors. And this is in accordance with what we know about the temperature sensation system in the Drosophila brain. Actually, cold and heat activate different neural centers in the fly brain, and different responses, individual responses of the flies correlate with different or activate different thermal receptors and regarding extreme hot or cold. So a hypothesis we may pose here is that navigation behaviors are rather controlled, but top-down integrated effects of temperature and through different genetic and neural mechanisms. Regarding our second hypothesis, we saw that in our population of study, the higher proportion of individuals of exploring the system were bold. And this is in accordance with a hypothetical curve we proposed about these proportions, right? If we consider these behaviors as a continuum, we can see different population patterns. In our population of isofemine lines, it's more similar to the red curve here. But time of the day was important, and this is in agreement with higher exploration and boldness during the morning peak of activity. but boldness is not necessarily the result of more flies exploring the system, like a density-dependent phenomenon or just underlying spontaneous locomotor activity. Some sex differences in the flies are explained by consistent but higher emigration activity in females than in males. And it's like males have a higher exploratory drive, but also sex differences in circadian activity on these flies too. These results may have some ecological and evolutionary significance in upper levels. For thermoregulatory behaviors may act as drivers or constraint for evolution on different niche axes. For example, in Anolis lizards, we have observed that thermoregulatory behaviors both constrain and promote physiological and morphological evolution, respectively. But in Drosophila, the question is if thermal boldness promote or constrain thermal physiology, and the answer is that we don't know. We observe a small reduction on thermal tolerance in thermally bold flies, but this needs further verification. But thermal exploration and boldness could be important at the edge of expanding ranges because individuals at invasion front of their distribution have higher dispersal, higher resting metabolic rate, and bold behavior. Perhaps this is a hypothesis. Hot thermally bold individuals may be favored under climate warming if thermal boldness in the heat opens some opportunities for individuals to exploit warmer microclimates, and if there are some foraging or mating advantages associated. In conclusion, so far we can conclude that the configuration of the thermal landscape modulates the heritability of navigation behaviors in fruit flies, that navigation behaviors through extremely hot or cold thermal environments are different contexts and elicit different responses, different genes, and different systems. But thermal boldness may not be a small fraction of individuals who display this within a population after all. So I would like to thank different institutions that supported this research of mine, my supervisors, both in Aarhus and in Brazil, the staff of the Department of Biology at Aarhus, especially Anne-Marie, my friends in Denmark, and my wife that has supported me in, always and especially during that pandemic time in which I conducted this research. And to all of you there in the audience, thank you very much for listening to me today.
So now we have some questions. And thank you very much, obviously, for your talk. The first question is, if the time of the day is important, does this behavior has a chronological base?
Probably that is a question also. I don't know the answer. The pattern is that there can be some effects. We know that flies have a daily activity rhythm that it differs between males and females, but in results that I didn't display, there is no correlation between the maximum capacity of flies to explore the system and the levels of thermal boldness that they exhibited. It's not that for more flies that explore, they tended to cross more. So perhaps there are some, there is some circadian component here affecting the exploratory drive of flies, but also some cognition processes here. And that is just a speculation at the moment because it's so much more what we don't know than what we actually know about this behavior.
So most probably a chronobiological study is one of the next steps.
Yeah, could be. could be. There are so many questions, and that's one of these. What this work is leading to.
Okay. Second is there a difference in the sensation of temperature in the brain of males and females?
No, that I am aware of. For some works conducted on these lines, there has not been any difference on that. What we know is that the thermal sensation system between the larvae and the adult phase work differently, but not regarding sexes in the adult form. So no, for as far as I know. I can be mistaken, of course, because there is so much Drosophila literature on this regard. But as far as I know, the differences are mostly related to ontogenetic stages.
The third question, why the behavior, even being conservative, when the animals decide to explore and experience, they appear to be boldness more than shy?
As again, I don't know. That's the most honest answer I can give. I mean, there is a bunch of literature that we can consider from, for example, the thermal performance curve, and also from some studies on behavioral ecology, which indicate that behavior may act as, I mean, thermoregulatory behaviors may act as constrainers for, as constraint for physiology and other ways. But in this sense, we would expect that behavior act as to conserve animal function, survival, even because we know that even subcritical temperature may affect reproduction rates and so on. But flies persist in their thermally bold behavior. I didn't display here, but there is we recorded these thermally bold behaviors, and there is one fly that we saw that was crossing multiple times this hot critical thermal zone without entering the food bottle. And that is weird, because we would expect that flies with hunger would tend to cross because they are hungry, and they would stay there, and that was what happened most of the time. But there was some flies that they crossed and stayed very close to the hot spot, but didn't enter the food bottle. So there is no intuitive reason that we as humans would expect for flies to do that. But that is also what we can see in human populations. There is a variation among risk-taking behaviors within human populations. And for some people, we could rational that there is no meaning, there is no sense to risk their lives doing, for example, extreme sports or whatever. But for others, it does have a reason. So I don't know how a fly think, but certainly there is so much more here. And when we see this persistence of flies to expose themselves to temperature that may kill them, we know that these thermally bold behaviors are controlled for something from above, from the fly brain and cognition and decision-making abilities, and not only for the bottom-up effects of how temperature affects the rate of movement or biochemical rates or metabolic rates and so on. So we don't know a lot of things.
Connecting with this part of the boldness, is there any general expected tendency in getting bolder as the animal get older? Is boldness related to mating behavior?
I don't know about the flies, but what occurs with some mammals and some reptiles, birds also, is that thermal evolved behavior occurs mainly in the juveniles instead of the adults. And these may lead to higher predation rates and things like that. So from this literature on behavioral ecology, we would expect that there is some sort of negative association between age and boldness, or considering boldness on different contexts, right? So that is what I am aware of, but I don't know about if that occurs also in flies, for example, in Drosophila.
And a final question. Do you have any feeling about how the reproductive state of males may affect their inclination to explore critical temperatures?
Well, so far all can be only a speculation, because our main, as Jesper explained yesterday, what we know about thermal behavior in Drosophila concerns thermal selection, right? And for example, the temperature that flies lay eggs and things like that. When there is a choice, when flies are presented a choice, parents may differ, and flies have the capacity, the cognition and behavioral capacity to choose whether going to a temperature that theoretically optimizes the development of those eggs and larvae. But I would say that if the fact females are carrying eggs, there would be a metabolic cost to these females, which are already starved by the protocol. And perhaps I would expect, for that reason, for higher metabolic demands, that flies could be that females could be more eager to cross to get food. But that would be something more associated to the females' needs and not necessarily to their offspring needs, to the immediate needs of the females. So that is my feeling, but again, it's just speculation at the time.
Well, thank you very much for our lecture and for the questions answered. We will have a 5-minute break for our last speaker. And yet again, thank you very much for the idea, Gustavo, and thank you very much for your presentation.
Thanks to you, and thanks to all, again, the Physio webinar staff and all the speakers that were so kind to accept to participate in this event. So thank you.
Okay. Now, we are very pleased to welcome Mitzy Becerras, a biologist by the National University of Colombia in Bogota, Colombia, and a PhD student in ecology at the Pennsylvania State University in Pennsylvania, USA. Her research is about physiological and behavioral mechanisms underlying parasite and host interaction in managed and natural ecosystem. Mitzy Becerras, thank you very much for your talk today, for your presence here today. After your talk, we will have 5 minutes to take questions from the audience.
Thank you so much for the invite. And thanks everyone for the curiosity on this project. So, muito obrigada. I changed a little bit of the title of the presentation. This has been a really crazy time here, very busy. So I'm sorry if I lost track of the title. I'm gonna talk about microbes and the impacts on their insect thermal tolerance. So highly dynamic environments like crops will undergo complex interactions among biotic and abiotic components. Here in this figure, Pocock used a network analysis to describe the interactions. The feedbacks are the black lines. Among herbivores, for example, you can see phloem feeders like aphids color-coded in blue, the pollen feeders color-coded in yellow, seed feeders in pink and orange, and some of their parasites. As we can see here, there are many interactions just at the herbivore level. However, the tradition...
Just a minute, I think you need to share your screen, your presentation. I think it's not sharing.
Really? Sorry for that.
Don't worry.
Now?
Sharing.
It's fine.
Yes.
So it's the screen. Okay. Can you see the Pocock figure with the network analysis? Okay.
Everything is fine.
Okay, so in this network analysis, we see a high level of complexity just at the herbivore level. However, our traditional and simplified view of the interactions among the biotic components in the ecosystem generally was ignoring the role of microbes, some of them pathogenic and other not. The immense complexity within the ecosystem expands across the spatial and time scales, with positive and negative interactions shaping the structure and dynamic of populations and communities. Abiotic and biotic factors can modify the outcome of insects' interactions with implications in the way in which they interact with one another and affecting the function and the structure of populations and communities. To understand the impacts of climate change on the physiological and behavioral adaptation of insects, we must study their response or their traits within a context, the context of their habitat and especially their ecological interactions, such as mutualist, commensalist, prey-predator, host-parasite interactions, because it will help us to understand the prevalence and the dispersal of infectious diseases, for example. Insects will experience extreme temperatures more frequently. Survival will become time-dependent during the highest temperatures. And physiological responses to increase intense heat waves will cause the reduction of energetic efficiency, oxidative stress, and survival mechanisms. Other behavioral responses that we can expect are shifts in daily, seasonal, and phenology, which combine with different migratory patterns and shifts in the species, will cause shifts in the species distribution. All these combined will likely result in change of ecological interactions. So today, I'm gonna give a little bit of background, and we are going to talk about 2 study cases in which viruses affect the thermal tolerance of their vectors and host, a plant host, and the impacts of fungus, a entomopathogenic fungus, on the behavior and the thermal tolerance of a prey and predator insects. So let's start with this. In the world, multiple stressors shape functions of the organism. The temperature has pervasive effects on the organisms on many levels. For example, the temperature directly affects the stability of proteins, the cellular function, and the physiological pathways. All these changes may affect behaviors like foraging, mating, habitat selection, with the impacts in their intra and interspecific interactions. It is critical to understand, I'm going to highlight this part, how organisms respond with their interactions, because the organisms, the species are not alone. For example, it has been increasingly shown that microbes, some of them pathogenic, may modify the physiology and behavioral parameters with impacts in the species fitness and niche size. Yet, there is much to be explored. And with climate crisis at most, more of these interactions or these dynamics are expected to change in unprecedented ways. So here in this beautiful but homogeneous environment, this wheat crop, fantastic interactions take place. One of these interactions is given by these 2 aphid species, Rhopalosiphum paddy and Rhopalosiphum matis. These species have as a host grasses. Paddy emerges first and arrives to the crop, followed by Rhopalosiphum matis. These insects, maybe you are familiar with them. I'm going to give a brief explanation of the system, and this is a very simple diagram of the system. So these insects are specialized phloem feeder, which means that they use their style, that is a mouth part that basically is like a needle that is injected in the phloem of the plant. They acquire the viruses. In this case, we are going to talk about barley yellow drive virus, that is a single RNA virus strain. There's a single RNA virus that has different strains. So this virus is acquired by the stylet and it moves around the digestive system till the mid-gut. In the mid-gut, it crosses the cells and then travels to the salivary gland, where in a species-specific manner, cross the ciliary cell of the salivary glands, which means that some that just by the strain BYDV-PAV can cross the salivary glands of paddy, and BYDV-RMB crosses the salivary gland of Rhopalosiphum matis. Additionally, these insects have a really interesting interaction because usually in the field, paddy is found on the upper part of the plant and matis is on the stem, so there is a spatial segregation. Well, these viruses are known to modify the behavior and the phytochemistry of the host plant, the behavior of the aphids and the phytochemistry of the host plant. So, which means that they are really important because they affect the response of the primary producers, plants. These changes that the viruses induce on the plant and the host have implications for disease transmission, as well as ecological interactions. For example, these pathogens directly manipulate the amount, the quality, and the availability of the resources on the plant that the plant offered to the aphid, like amino acids, phytosterols, sugars, and as well as the derived chemical cues that attract and retain the insects. However, few studies have explored the direct effects of viruses on thermal biology of hosts and plants, of the host plants and the vectors, and the broader implications of these pathogen-induced changes in the host and vector phenotype. So right now, I'm going to talk a little bit about thermal regulation. And well, I know that the audience is very familiar with the subjects, but here with this picture, I just want to illustrate that this is a environment, a very heterogeneous, small microhabitat, a apple leaf that offers this variety of temperatures where the insects move around trying to find a temperature that allows them to match their optimal temperature. So here in this following slide, we are going to talk about a hypothetical environment or hypothetical habitat where we have a cold specialist and a warm specialist. So here in these unfavorable conditions, the cold specialists move around trying to find a temperature that allows it to survive. It could avoid these hard or harm conditions by switching resources and altering its behavior. But what happens if there is an organism like this warm specialist that has an enhanced tolerance? The next question is, are there any biotic interactions that are altering or inducing this? And here is where we were very interested, because this system, PADI and its strain BYDV PAB and MADIS and the strain BYDV RMB are spatially isolated. There could be other parameters on the host plant that are driving these interactions. So we hypothesize that the virus alters the thermal biology of the vectors, and we expect that the virus induces changes in the need, size, and the strength of the ecological interactions between the vectors. With the help of my amazing students, Catherine, David, Martin, and Kevin, we conducted several experiments where we explored the effects of viral infection on the thermal biology of PADI and MADIS, and the resulting effects of their interactions with one another. We conducted laboratory and field experiments using different strains of BYDV, as I mentioned before. We used novel sensors to characterize the surface temperature of the plants, the spatial distribution of the aphids in the field. We conducted some physiological experiments where we measured the heat tolerance of the aphids, which were infected and virus-free. And then we constructed transcriptors to identify upregulated genes under their outstanding insect response. And we measured the expression levels of these genes. So as a result of our measurements in the field using the thermal camera, we found that there is a thermal gradient along the host plant. As you can see, there is a blue area here that corresponds to the stem, and this top part corresponds to the spike or the flag leaf. I'm sorry. And there is a big difference in temperature between the stem and the flag. Then we moved to conduct our experiments in the climate-controlled chambers, and we use plant surface temperature as our response here. Here we infected our plants with the different strains and compared them to a control plant. We found that the infection by BYDV PAV increased the temperature of both stem and leaves by an average of 2 degrees to 3 degrees Celsius. However, no similar effects were observed for the infection by BYDV RMB. In the field, we manipulated the presence of both aphids and the virus strains. Here in this graph, you can see in the y-axis the temperature on the plant of each aphid location, and 2 treatments, Rofalucifum PADI alone, Rofalucifum MADIS co-occurring with PADI. Here, I'm just gonna give the details for PADI. We observed that PADI preferred the stem that correspond to the cooler areas of the plants. Here, the average temperature is about 18 degrees Celsius. When PADI was infected, alone, this preference didn't vary. However, when paddy co-occurred with maize, paddy moved to the upper parts of the plant. And this was modified. The place where paddy was living when it was infected was in the upper parts of the plants that are even warmer than the previous ones. Then we assess the upper temperature limit for locomotion, the Ctmax, of both insects when they were healthy or virus-free and infected by manipulating the temperature, as you are very familiar with. This figure shows the Ctmax in the y-axis and the treatments in the x-axis. The average Ctmax of virus-free and virus-infected paddy, we found that Ctmax of paddy infected was 8 degrees Celsius warmer than virus-free individuals. In contrast, as you can see here in the panel B, BYDV RMV did not significantly affect the Ctmax of maize. To explore the potential mechanisms underlying the increased heat tolerance in infected paddy, we extracted messenger RNA from virus-free and viruliferous aphids at room and under heat stress given by the Ctmax, and in collaboration with Juan Ragosa, we constructed transcriptomes for each treatment condition using Illumina sequence and compared them to determine which genes were up or down regulated. The analysis identified more than 40 genes, and 3 of them encode heat shock proteins that are associated with tolerance to heat or stress. Then we conducted quantitative real-time PCR to measure the expression level of these 3 heat shock protein genes, and we found that expression levels were significantly higher in the condition infected and exposed to Ctmax. As a conclusion, we can say that viral infection increased the plant surface temperature by 2 degrees Celsius and confers heat tolerance to its vector paddy, while no similar effects were apparent for BYDV RMV infected individuals or infected maize. This enhanced thermal tolerance is associated with differential upregulation of 3 heat shock protein genes, and the viral infection allowed paddy to exploit formerly unsuitable regions of the host plant. As a main take-home message, thereby we can say that the virus expand the fundamental thermal niche of its vector. And furthermore, the viral infection led to the large increases in the fecundity and lifespans of aphids under heat stress conditions, especially for paddy, while ameliorating the negative effects of interspecific competition. Now, I'm going to briefly talk about the second case of a study that was the impacts of a ethnopathogenic fungus on the thermal tolerance and behavior of prey and predatory insect species. Now we know that these 2 viruses strains alter the host plant volatile profiles, the nutritional content, the thermal tolerance of one of the vectors, but do other microbes alter the performance of other species across the trophic levels? And if they do, how? Building upon these findings, we are expanding our research to other model systems like the entomopathogenic fungus Beauveria bassiana and the aphid Acyrtosiphum pisum and the predator Hippodamia convergence. Here, well, you see another cartoon of Critidia mellificae and Osmia cornifrons that are our current experiments, but I'm not going to talk about them. I will be running out of the time. So the incidence of extreme thermal events may alter the organismal function, leading to changes in trophic cascades. Although changes in mean temperature do not seem significant, small shifts mean big changes in extreme temperatures. Research in the impacts of extreme temperature has been gaining attention because the increase, the frequency and the intensity of these heat waves is increasing. For example, extreme temperatures are important for interactions like rate attack. The extreme temperatures also affect decrease the prey consumption across trophic levels. It has been shown, these changes have been shown in rotifers and dragonfly larvae and other species like and other attacks like fishes. Extreme temperature can also affect the survival and the viruses of fungal pathogens and other aspects of the pathogen-insect interaction, which led us to this project. So for this experiment, we moved our entire setup to the field. We grew our aphids and beetles in the field and sprayed fava bean plants with 2 different concentrations of Beauveria bassiana. We basically found in this figure the x-axis represent the CT max and the y-axis represent the CT max and the x-axis the treatments, healthy individuals and individuals exposed to 2 different fungal loads. The figure A shows that fungal infections reduce the heat tolerance in assertive Physium by 7 degrees Celsius. And in hypothermia conversion, the CT max was reduced by 4 degrees Celsius regardless of the pathogen load compared to the healthy ones. Regarding the CT mean, in the panel A, you can see that the fungal infection didn't significantly affect the CT mean of Ascirtosiphon pisum. However, predator beetles' ability to withstand cold was reduced according to the pathogen load. And then, using the famous experimental setup that was developed by Professor Carlos and collaborators, we expose our individuals, the healthy and the infected ones, using groups of 10 individuals in the home bottle, and we allow them to freely move across the T tube for 1 hour. Then we may, and in each side of the bottles, or the extremes, we had different stimuli that were leaves or were aphids for the leaves for the plant leaves for the aphids and aphids for the predator beetles as a stimuli for movement for these individuals. Here, we found that, interestingly, the individuals that were healthy were very bold. 60% of the population decide to cross the warm extreme zone, rather than 40% the cold extreme zone, but there were clear decisions. These vary when the insects were infected, as you can see here in this picture. We observe a similar trend for the predator beetle. Here, we demonstrate that the infection shifts behavioral response for both predator and prey in ways that reduce the exposure to heat, which challenges the notion of fungal infection in insects that are expected to compensate the infection with behavioral fever. Later, we measured the ATP levels of the insects that were exposed to each condition, and we found an interesting pattern, a high energetic coast associated with short cold exposure. As a conclusion, we can say that fungal infection reduced the CT max of both aphid and predator beetles, as well as the CT mean in the beetles. The infection modified the tendency or boldness of aphids and predator beetles to cross either warm or cold extreme zones. And the ATP levels increased with pathogen infection in both insect species. And the highest ATP levels were found in individuals that crossed the cold zone. Collectively, sorry, our study demonstrated that the fungal infection narrowed the thermal tolerance range and inhibited thermal boldness behavior to cross extreme temperatures. We challenge the concept of behavioral fever here. As a conclusion, we can say that these two experiments have been an amazing systems and have allowed us to observe and describe very interesting interactions between microbes, insects, and plants, and the way in which they respond to thermal stress. So, muito obrigada pela atenção nesta palestra e o interesse e a curiosidade. Thank you so much. I will be happy to take any questions. And some acknowledgements to many people, students, funding agency, and collaborators, Professor Carlos, Gustavo, Jesper, Volker, Consuelo, and many people that were contributing in different ways to these experiments. Thanks. I can hear.
Okay, thank you very much for your presentation. And now we are going to take some questions. The first one is, do virus infections affect cells so they can stimulate the AT and HSPS production?
Well, the infection of the virus is something that is very interesting because most of the research attention has been paid on the chemical ecological side. So we know very little from the interaction of the virus on the vector. I can say that from a experiment conducted by Jesper Jansen, like, I don't remember the date, but it was many years ago, he demonstrated that there was, using in vitro experiments, that there was a level of affinity of the heat shock proteins, like GROEL protein, and the viruses. So we make inferences like this could be a mechanism associated with this response, but we didn't conduct an experiment that allows us to demonstrate the level of affinity or so in the aphid. That is the closest answer.
Okay. Connecting with virus, about the result of the virus enhancing the heat tolerance. Oops. One more question. Heat tolerance of the host. Do you consider that infection does not, about the result of the virus enhancing the heat tolerance of the host, in case the vector, I think, as well. Do you consider that infection does not impose a trade-off to the heat tolerance? If so, why?
Well, in this system, we can make different inferences about, for example, I see that the question is about ectothermic vertebrates. And so, well.
No, that's the third question.
Okay, so for this system, there is a coevolution between the vector and the viruses in ways that they have coevolved in the ways that benefit the dispersal of the insect as well as the virus. And well, usually this is a very complex system in which it seems that the virus manipulate the entire system in ways that make it, for example, plants that are infected with the virus have more sugars and phytosterols and amino acids that help the vector in different ways. For example, the vectors that reach these plants, they have higher offspring. And with higher offspring, the density of these individuals in the plant induce the wing form. So these insects with flies are the ones that are in charge of traveling further distance and disperse the virus. So here there are more benefits than negative sides. other experiments, we found that even if the insects are infected, but are exposed to the Ctmax, so the thermal stress, let's call it, there is a reduction in the offspring, but still the increases in the offspring are huge. I didn't want to show the results of this, of the offspring and survival, because it was too much detail, but the increments in the offspring when the insects are infected is about 200% more. So you can see, for example, naturally, they lay 3 or 4 aphids, but when they are infected, they lay 8, 10. So the difference per individual, per capita. So in this case, it's mainly positive things for the aphid, not for the plant.
Yeah. Now the second one, does insects exhibit behavior fever as seen in ectothermic vertebrates? Can the change in the thermal tolerance also be a side effect of fever?
It could be, yes. We just make some inferences about the response of the insect because it was very interesting for us. Usually, well, the literature in grasshoppers in Locusta, and when they were infected with other fungi, with other entomopathogenic fungi, show that if you expose the insects in an arena that has different warm temperatures, but just warm temperatures, insects were to the highest temperature. But now with this setup where the insect can move live free, we show the first time to our knowledge that insects could make other decisions. And it was interesting that most of them prefer to stay in the bottle. They were not bold and cruising. So it also has, like it would be more, that's something that we would expect because there is an energetic depletion with infection. There are other physiological pathways that are affected with infection as well. And we also could say that here at this point, this experiment was very manipulated, like we use individuals of the same age, we spray them with the fungus and they were infected for 2 or 3 days and that's it. So the changes were not maybe, well, the experimental conditions were a little bit different from the experiments that were conducted before with Locusta and so on.
Okay, the fourth question is, in a real scenario, could the use of biological fungal control in Bioveneria bassiana in farming directly affect the survival of aphid predatory insects like ladybugs?
Yes, that's true. In this experiment, we also measured the lifespan, and most of them were dead. So, yeah, in the real scenario, the direct effect definitely affects survival. It killed them.
And the fifth and last question: do you know what different costs impose a viral versus fungal infection on your study host species? I ask for the different effects of these infection on host thermal biology.
Well, some of the differential costs come from the evolutionary history. You know, we just mentioned that, for example, in the case of the virus and the vector, there is a coevolutionary history behind of them, in which they both receive benefits from their interactions. So maybe under those conditions, we can infer that, or we could expect in other systems, if we project that maybe other aphids or other insects, not in fact, in white flies, Bemisia tabaci, and another Lepidoptera that were infected, there are different responses with. So Bemisia, I don't remember well, but one of these species had increased heat tolerance, and the other was infected with the virus, had lower heat tolerance. So it depends on the system as well. Could be like related with the level of association or affinity of the virus with heat shock proteins, as Jensen demonstrated. But for the case of fungal infection, it would be different. It also will be related with the level of, I would say, the immunoresponse of the aphids or the other insects like locusta, or maybe other insects. Like, for example, here right now, we have an invasive species that is called the spotted lanternfly, and they sprayed it, and with Beauveria bassiana, and there is no significant effect on mortality. So it also depends, it's a species dependent.
Okay, thank you very much for your presentation, Mitzy. It was super interesting. I was so deeply interested about your job, and I think our audience was as well. And we are very thankful for you here.
Thanks for inviting me. It was a great opportunity to share this work.
So right now, we have an announcement from Gustavo Agudelo. So I would like to give him the word.
Thanks, Maiara. Yes, I would like to thank again all speakers for this opportunity to have conducted this Physio webinar, the staff, and also would like to announce a follow-up event that is being shown at your screens now. This Physio webinar was dealt mostly with the theory and data, but it is also important to know how this data is gathered, right? So a follow-up event on these topics, it's a specialized workshop on methods and approaches to quantify some thermal traits. This workshop will occur on the 31st of May. It would be a virtual workshop that will be announced and disseminated within the University of São Paulo. But if there are some people in the audience that is not from the University of São Paulo and that is interested to join this event, please send me an email. There is the information here in the screen. Send me an email with your interest, because the idea is that people that are interested in participating will join this workshop. And just for you to know, some of the topics we will cover, it's about describing this thermal decision system that we developed to study thermal boldness, right? So I will describe the behind the scenes of how we constructed this system and the particularities of the experiments. There will also be some topic about the versatility of experimental approaches in comparative thermal biology, the importance of natural history of species to cover and to modify experimental settings, right? And the final topic would be about the particularities of a quantitative genetic study, right? So taking thermal biology as and thermal traits specifically as a context, we will see and learn how to perform a quantitative genetic study, how to estimate parameters like variance components, heritabilities, evolvabilities, and all of that, right? So that is my invitation. For people from the University of São Paulo, wait for the email to get. And for people that are interested from other institutions, please write me an email and send and let me know your interest, and I will consider you too. Thank you.
So today we end our talks, and many thanks again to the speakers for taking time to join us and for the wonderful conversations and discussions. I would also like to thank you everyone who watched our broadcast, as well as you who will be able to watch in the future when it is available in our YouTube channel. Be sure to follow us, our channel and our social media for staying tuned of upcoming events of the Physio webinar and Physio webinar spin-offs in the coming years, with different researchers from around the world presenting their researches on animal physiology and their intersection with different areas of research. I would like to, as well, to thank you our group, our team. I would like yet again to extend thank you for Gustavo for the idea, and to everyone that were here to accompany us. So thank you very much, and see you.